Good afternoon. Welcome to BioCom 2021, the June edition. So I'm Adam Cooper, your president of the BioCommunications Association. Welcome, everybody. It is my pleasure to introduce to you Jamie Hayden, our vice president. And I don't actually know your title at Wistar. You can tell your title there. And he is going to talk about confocal microscopy and how he teaches it to his people. And what we're going to learn a lot about this technology today. So let's give Jamie a virtual round of applause, and we will pass it over to Jamie. Go ahead, you're on.
Thank you, Adam. That was a lovely thing. My official title is the Managing Director of the Imaging Shared Resource here at the Wistar Institute. So welcome to Wistar in Philadelphia, PA, and welcome to my lab, or at least this little corner of it. Happy to do this today. This is something that, when I realized we were doing all this virtual stuff, it kind of gives us an opportunity to look at something we haven't been able to look at before. So this will give you a flavor, especially if you are not familiar with more modern approaches to microscopy, but hopefully I'll give you some ideas that sort of bridge the gap as well. So there's a lot that we're going to be covering. We're calling this a workshop mainly because there is so much to get through. If I can just make sure a quick note on there. Okay, so my first question that I was asked when we wanted to get started on this is, what exactly is a core facility, which is what we do. So shared resource or core facility is a common name. And the best way to explain what that is to think about how much these instruments cost. Back in the old days when I had darker hair and was just learning microscopes at the foot of people like Martin Scott and people like that, you could get a good high-end research-grade microscope for $50,000 to $100,000, somewhere in that range. It was analog, worked with film, had all kinds of bells and whistles, and that was great. Pretty expensive, but if that was something that your lab did a lot of, that was useful. Well, fast forward to now, and the instrument that we're going to be looking at today, when we purchased this, it was $550,000, and the more recent ones that are out there are in the range of 7, 8, 900,000 or more, depending on how many bells and whistles you put on those. So obviously, every lab that wants one of these things really can't afford to maintain one of them, and also can't maintain the expertise necessarily to teach everybody going forward. So that's sort of where the cores come in. The whole idea is that an institute like Wistar, and at Wistar, we do a lot of basic cancer research, immunology, vaccine development, that sort of thing. If the Institute supports a facility like ours, we get to buy all the high-end equipment, learn how to use it, and then teach everyone in the Institute that needs to use it and provide the opportunity for them to come down. So this training aspect of it is something that I do an awful lot of whenever we get anything from a grad student to a postdoc to a PI in here that wants to learn how to use it. We go through this process. Several years ago now, like 2 years ago, before COVID, my training regimen for the confocal took 3 days. We were dealing with three-hour sessions, hopefully 3 days in a row, but at least very close to it. And what we did on the first day was I sort of sat here and we went through the entire soup to nuts process with you sitting by my side and taking notes and doing all this wonderful stuff. By the end of that first day, most people were very overwhelmed and were thinking, why did I get into this in the first place? They come back the second day and we switch seats. On the second day, they get all the hands-on. I sit on my hands and they're going to be then running everything from their end and with me telling them what to do. But we basically kind of repeat everything we did the first day and then add a little bit more to it. By the time we get to the third day, they're getting much more comfortable with the system. And we get into things like 3D and the specific requirements for their particular experiments. So that's why it took as long as it did. Well, a year and a half ago, all that changed. all of a sudden, overnight, we couldn't sit side by side with people anymore. And if you had asked me before to do training on a confocal remotely, I would have said you were nuts. But we've managed to come up with a way to do that. So what you're going to be seeing here today is actually most of what our first day's training covers. Now, in order to be able to do this, we've actually made things a lot more efficient. We started with the training videos. So hopefully you all had an opportunity to take a look at the linked training videos for this program. If you didn't, you might want to go back after this to follow up on them. But those start with some of the hardware things, how to get things turned on, which objectives to pick, how to put a slide on the stage, all that kind of stuff. So I'm going to kind of gloss over a lot of that in my talk today. But those training videos gave that first bit of what we usually did in person. And then once we got past that first part, a lot of the action, if you will, really took place on the screen, on the monitor. And so once somebody has a slide on the stage, I can be in my office. We can be sharing screens. I can be looking exactly at what they're looking at. And with Windows 10, we can share a mouse, we can both have a mouse. So I can point at things, I can click on things. And that's the way the training gets done. So after I stop with this initial part, what I'm going to be doing is sharing my screen with you. You're going to see the software, and we're going to kind of go through the entire process. So what you have to remember from the beginning here is that confocal is a fluorescence technique. So all the light that you're going to be getting in your microscopy approach is from fluorescence. Now, fluorescence can come from fluorescent proteins, which are integrated into the cells. It can come from some sort of immunolabeling, where you're taking a fluorophore and attaching it to a specific antigen and doing an antibody stain to go along with it. So you're picking a stain to go along with other fluorescent objects that you're working with, any combination of these things. So really, to begin with, as with any good photography process, what you really need to know from the start is everything. You have to understand the science. You have to know what it is that the researchers are looking for. You have to understand how your equipment works inside and out, because when they come up with something that doesn't quite work right this way, you've got to know that you can do it that way and come up with another approach to it. You need to be able to plan the experiment in a way that the answer that it gives you is valid. Sometimes people come in thinking that they're going to get an answer that actually isn't even valid. So it's always best if they speak with us first to design their experiments. And in that process, we'll go over things like what fluorophores are going to work best together and how to avoid bleed through issues and stuff like that we'll be talking about, and I'll be showing you a little bit later. So to that end, what are we going to be working with today? What I have is a training slide, and in this training slide, it has bovine endothelial cells, which are essentially like a type of fibroblast, if you will. They're labeled with 3 different fluorophores: Dappy, which is used to label DNA and is most commonly seen in the nucleus. So you're going to see the nucleus. Then there is a Alexa 488 dye, which is attached to fluorine, which attaches to actin. And so we're going to see actin filaments using the Alexa 488 dye, which traditionally would be a green color, and the Dappy would be traditionally a blue color. And then we also have the mitochondria labeled with something called mitotracker red, which will only attach to mitochondria. So we'll see the mitochondria as well. So it's a triple labeled slide, and that's what we're going to be looking at. So I think without any further ado, what I'm going to do is share my screen with you, and we're going to get right into the software. So you can see what I get to play with all the time. All right, now, as I said, I'm assuming that you have had the opportunity to watch my videos in advance. And so where we are in the process then is we have started everything up already. We've taken a slide, we put it on the stage. I've already focused and found a cell just to get started. What we're really going to be talking about today is all the options that we have in the software. The reality is that when you're doing confocal, it is a balancing act. You're going to choose this way versus that way. If you ask me, how do I make that image brighter? I could tell you 6 different ways to make it brighter, but every way that you pick comes at a cost. And what you have to decide from the scientific point of view and from your experiment point of view is what's going to be the best combination of settings to give me not only the data that I'm looking for, but also, if you're like me, a good looking image. Sometimes you can't get a good looking image, but you get great data, and sometimes it's the other way around. So sometimes it just kind of depends on what your purpose is there. So you see on here, it's a little bit like a 747, and I do apologize for the fact that the text is extremely small. If you can't see it, don't worry too much about it. I'm going to be reading it out to you so that you'll know what we're looking at. Hopefully you can see I'm moving the mouse around in the top left quadrant, so I'll be pointing at things with the mouse while I'm discussing our options. And you can just see what the process is to go through this. So we have our 3 dyes, and the very first thing that we have to do is we have to go up to the top left to the configuration tab, and we have to make sure that we select the lasers that we're going to work with. So first thing I got to do is sort of explain a little bit about fluorescence. Traditionally, fluorescence is done with a microscope that incorporates filter tubes. And so what you're doing is you're taking a bright white light source, which is going to go through an excitation filter. Let's call it a blue excitation filter with a particular set of wavelengths. So all the light that's coming out the other side is now blue. That blue light is going to then hit a dichroic mirror, which is going to send it down to the sample. And once that blue light, that excitation light, hits the sample, if it is, for instance, Alexa 488 or GFP or one of those green type fluorophores, is going to emit a green color. So that green light is going to come back up towards that dichroic mirror. The green is going to make it through the dichroic mirror, but any blue light is going to be reflected away. That's why it's called dichroic. It lets through certain wavelengths and it reflects other wavelengths. And in the end, that green light then passes all the way through an emission filter back up to our camera. So if you understand that basic concept of fluorescence, you can begin to see the beginning part, at least, of confocal. So instead of using a white light source, we're using lasers. So we don't need to have an excitation filter anymore because a laser is a monochromatic light source. So you're just going to pick a laser with the wavelength that matches the excitation that you need for your fluorophore. So if you look at my choices here, you can see that there are 5 options. 4 of them are black right now. The argon one is turned on. We'll get to that in a second. And the first one is the 405 nanometer diode laser. So the 405 is the one that we're going to be using to excite Dappy. Interesting little thing for most of us photographers out there is that the typical excitation for something like Dappy should be closer to 365 nanometers, which you might recognize as being ultraviolet. The problem with trying to excite with ultraviolet is that almost every optical surface in that microscope is coated with an anti-reflection coating which blocks ultraviolet light. So guess what? We can't use ultraviolet. So they discovered a while ago that a 405 nanometer laser will get through, and if it's bright enough, it will excite the Dappy, which is a bright fluorophore to start with. So we're going to turn on our 405 laser. The argon, I've already turned on, mainly because we have an issue with the fans and heat in here. The argon puts off a lot of heat. The argon actually has 5 different laser lines to it. So I actually have 9 different lines I can work with. To begin with, the argon, even though it's turned on, is not actually on yet. It is the power switched on, but it's at a 0%. I'm going to be setting this to 25% of its full power. At full power, it's actually a very strong laser. It's a 60 milliwatt laser, which would pretty much burn through anything that we're trying to look at. When we're imaging, we actually want to keep our the intensity of our laser under a milliwatt. And so we don't need to start with anything anywhere near 60 milliwatts. So I'm going to only bring it up to 25% to start with, just so that we don't keep it at 100% all the time. Now, the other laser I'm going to be turning on is the 561 nanometer laser, and that's going to be for our mita tracker red in this particular case. Now, when I'm doing the training, you'll notice there's a whole bunch of other little buttons over onto the left. And those of you who do training with anyone will recognize that most people will look and they'll say, oh, goodness, what does settings do? What does control panel do? And they'll go click, 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 click. Unfortunately, there are many things underneath these buttons that if clicked on, stay clicked and kind of messes it up not only for you, but for everybody else. So what I tell everyone during training is there's only two things under this configuration tab I want you to work with. One is to turn on your lasers. Two is to go to the settings button here. And there's only one thing here that you're going to click on. So down here at the bottom, we have resolution. So without getting into a long discussion about resolution, because that term is used very loosely here because there's a lot of different definitions for it. Resolution in this case refers to bit depth. So by default, when you turn on the system, it's going to be capturing at 8 bits. And most people using images from a confocal actually want quantitative values, quantitative data. There are two rules when you're shooting for quantitation. One is use high bit depth, and two is make sure that you don't oversaturate any pixels. Otherwise, it invalidates the results that you get. So unfortunately, I cannot change this. It's always going to start at 8 bits. Now, 8 bits is 256 levels of gray. Your eye cannot even see that much. The screen that you're looking at cannot even show you that much. But the detectors on this microscope are actually 12-bit detectors. So what we're going to do is we're going to select 12 bits in the resolution so that we're capturing at the full bit resolution of this particular microscope. And by the way, this is a Leica SP5 laser scanning confocal microscope. Next month, we're going to be working with a newer version, the SP8. And just to give you an idea, we got this thing about 9 years ago, the SP5. The SP8 came out about a year later, and they're already out with a newer version of it now. So we're about 3 generations behind looking at the technology I'm giving you today. But this is still my basic training process. All right, once you've gone through the lasers and the settings, get out of here. We do not want you to click on anything else. So we go back to the Acquire tab, and this is where, as I said, it looks a bit like a 747. So we're going to go through all of our options and talk about how we make the choices that we make before we get anywhere near looking at an image. So to begin with, let's go to the top left and work our way down. The first top left is acquisition mode. It says XYZ, which is the way we're going to be shooting. Just for interest, there's a few other things that you can pick here, including things like XYZT, which would be time-lapse. So that would be 3D time-lapse. But if you have something like XYZ, let me ask you, what is XZY? Because that's an option as well. It has to do with the way the scanning takes place. So XYZ is going to scan in your traditional focusing up to down mode. XZY focuses side to side. It's really crazy stuff. I've actually never had to use that mode, but I know it's there. Okay, the next button here is the sequence button. And actually what I should do is kind of reset this back to the way it looks when you first turn on the software. One of the problems with some of this more advanced software is you need to know what you don't know yet, which makes life a little confusing. So if there's not a box there, you need to know that you need to turn on that box, which is, can be difficult. So the sequence box is actually this little button here in the top left that says sequence, and it's not turned on to start with. But I will tell you right now that if you are using Dappy, which we are, you will be doing things in sequence. So I'm going to turn that on. Now, as I said, with sequence, or I didn't say this yet, but there's basically 2 ways to image with a confocal. You can do things in sequence or you can do things simultaneously. Traditionally, we think of doing it simultaneously, and when we first see an image, that's the way we're going to look at it. But you're going to discover that you're going to run into some problems that way, and so the sequential scan is usually the more common approach. So we're going to need this box down here later. So we've turned it on. The next 2 buttons in here have to do with if we are doing any kind of scaling, like time scale, not time scaling, scanning, where you're doing multiple images and putting them together. Or if you're picking one spot and another spot and going back to the first spot again. So if the stage is initialized, it lets us go back and forth around there. In the videos, I told you never initialize the stage, right? Well, if we didn't initialize the stage, these buttons would be grayed out. But in this case, I did initialize the stage so we can see them. I have a little saying in basically everything to do with most types of microscopy and photography. There's a lot of swear words out there in the world, and in English, most of those swear words are 4 letters. Well, I have one for you, AUTO. To me, auto is a 4-letter word. It's a bad thing. So anytime you see something that says auto on it, so if I mouse over this button in the top right, it says show autofocus panel, get away. There's nothing good under that button. Basically, the reason I'm so much against auto is because systems like this were designed because they understand in some respects that some of the users are not going to really understand it very well. And so they want to put in buttons that make things automated. So you don't have to make those choices yourself. You can let the computer pick those choices. Well, I'll tell you right now, I don't agree with a lot of the choices that this thing makes. And so if I wanted to do it, I'm not going to get the consistent same results every time. So I want to make sure I'm not using auto. So anything with auto, I'm going to turn off. Now, the next little sliver right here that says XY, it's hard to see, is actually one of the more important boxes on this software. So I'm going to open it up with a little arrow there. You can see there's a lot more in here. And this is actually where we're going to have to spend a little bit of time right now. So to begin with, when you first turn it on, it's going to look like this. You're going to take a look here at format first. So when we image with the confocal, we're usually taking square images. And the format here is telling me 512 by 512. So roughly that's telling me 512 lines down, 512 pixels across. That's what makes up my image. Generally, what we're going to be doing as a sort of rule of thumb is you use 512 by 512 in live mode when you're looking around, focusing, scanning, things like that. And then you're going to switch to 1024 by 1024 when you actually take your images. So we're going to leave the format in that mode for right now. Now, underneath this is speed. So if I click on speed, you'll see there's a bunch of choices. They start at the bottom at 10 hertz and they go up to 14 hertz. And then they give us a choice here of more. By default, it starts off at 400 hertz. Well, what does all that mean? So hertz is lines per second. So if I'm talking about 512 lines per second and I set this to 10 hertz, it's going to take quite a while to get through 512 lines. So that means my scan speed is going to be incredibly low. So obviously that extreme is not particularly good for us. But why would we even want to do it in the first place? Well, in general, the lower the scan speed, the less noisy your image is going to be. So the better quality you're going to get out of your image. So the slower it goes, the better the quality. Well, if you like sitting here all day, that's great. So I'd rather go a little bit faster and deal with that noise issue later. So how about going up to 1400 hertz? Well, the problem with going to 1400 hertz is that mechanically there's problems with the system where if I go to 1400 hertz, I can't get the full field of view out of my system. So if I have a 63x objective in there and I want to get, maybe 10 cells in my field of view, I am going to be limited to a 6x magnification on top of that if I go to 1400 hertz. So that doesn't do us any good. So what am I going to set it at? Well, I'm actually going to set it at 600 hertz. So I go to the more setting and I come down here and I put in 600. And the reason that I'm going to 600 is that that's going to give me the fastest speed that I can go with and still keep my zoom level down to where I can see my entire field of view, or a 1x field of view. As soon as I go above 600 on this particular scope, I'm limiting my field of view, and I don't want to do that. So we're going with speed, and keep in mind now that that is 50% faster than the default. The default said 400 hertz. We're doing 600 hertz, so we're going 50% faster. It does make it a slight bit noisier, but in my experience, it's nothing that we can't handle. So we're going to go with that. All right, the next thing, there's two things I'm going to talk about here. The first one we'll talk about is bidirectional X. So I'm going to turn on bidirectional X, which is that one right there, top left. So a scanning laser works a lot like an old typewriter. So if you think about it, what happens at the beginning is you start typing and you're going to go left to right, scan, scan, scan until you get to the end. And then you're going to hit the return and it goes back to the beginning and it goes down a level. And it then scans in the same direction until it goes to the other end, and then it returns and goes back. So that is actually unidirectional scanning. It's going left to right and then returning and then scanning left to right again. And so the question is, why do we do it only left to right? Why don't we do it left to right and then right to left and then left to right? Wouldn't that speed things up? Well, yes, it would. So we're going to do that. That's what bidirectional does. I had an old confocal before that had this option. Before this one, we had the SP2. That particular system was horrible with bidirectional. And so I never used it on that particular system. But on this system, it's very good. So we turn it on, and now we just sped everything up by 100% now. It's going to go twice as fast as it was before. So that's a good thing. All right, now directly above bidirectional, we get to pinhole, and this is where we're going to have to spend a few minutes to explain exactly how confocal works. So I'm going to turn on pinhole, and you're going to see that down here a few things showed up. What I'm going to first do with this software is I'm going to change the unit. So the unit right now is in microns. So this is actually the size of the pinhole. We'll talk about what that is in a second. But knowing the size of the pinhole doesn't do me a lot of good. What I really want is as a standardized reference. So I'm going to change the microns to AU, which stands for area units. And that's going to give my pinhole an area unit of 1. So 1 is my default standard setting, and it's going to be pretty much across all confocals. You're going to start with 1 area unit. At any time, if I want to change that amount, I can get back to it. So I have this little slider here. I can move that. I can change the amount of the area unit. It's now 2.83. If I want to go back to 1 area unit, I can just hit that area 1 button right there and go back to it. So it gets me back very quickly. All right, so let me kind of explain what the pinhole does and how it works. So to do that, I am going to have to come to my PowerPoint slide. Okay, so this is the microscope that we are working with right now. If you had a slightly different angle than the camera on me has, this is what you're looking at. Over to the left, you have your basic inverted scope inside an incubation chamber, and over to the right, I'm sitting in front of that screen and talking to you. All right, now inside this fairly basic microscope, there is the stuff that makes it a confocal. So we're going to go a little diagrammatic here and really kind of talk about it. So if you look here, what I have is going to turn into a very simplified ray diagram. For any of you who are really good physicists, I apologize in advance. I do this to get people to understand the concept more than the specifics. So at the bottom, we have our sample. And you're looking at this, we got like a brown cell with a blue nucleus in the middle of it. And I'm going to start by explaining to you the first very big mistake that I did when I first started working with a confocal. I think most of us understand that confocal is a process to essentially visually section through a cell, top to bottom, without actually cutting through it. You're focusing at different planes and you're throwing out the out of focus light. And so how do we do that? That's what we're about to talk about. But my understanding of that was a bit limited because I thought, okay, if I'm only focused in one plane, I'm only illuminating that plane. One of the biggest problems in fluorescence of any type is bleaching or fading or quenching. I'm using those terms interchangeably. But the idea is that the more you hit your sample with light, the more it's going to fade on. So we really need to control that fading. It's a big problem. Now, if I'm only looking at one plane, I'm only going to be hitting that plane with the light, so that's the only one that's fading, right? Wrong. As it turns out, that's completely wrong. What really happens is we are exciting the entire volume of the cell. And so what we're going to do is we're going to look at where those three dots are, the red, the green, and the blue dots. Those are in three different Z locations in the cell, going from top to bottom. And if you looked at these straight from the top, they'd be one on top of each other. So you wouldn't be able to tell that they were in different places. But from the side, we can. So the blue dot is on the cell membrane, the red dot is in the middle of the nucleus, and the green dot is in the cytoplasm on the other side of the nucleus. We are focused right at the middle one. Our plane of focus is on that red dot. Now remember, any type of lens only focuses at one plane at any given time. There is no such thing as depth of field when you're dealing with microscopes. You only have that one plane of focus. So what we're actually going to do when we focus is that we move the cell up and down through that plane of focus that we have. But for right now, let's take a look at what happens with that light. If we are focused there and we're exciting that red dot there, where is that going to go? Now above here, you have my representation of two lenses, right? Because throughout this entire confocal process, there's a whole bunch of lenses, and the light from that spot is going to cross over and go past itself many times before it gets to the camera up at the top. Where somewhere in between all this, we are going to place a pinhole. And that pinhole is going to be at the confocal plane to where we're in focus. So hey, there's where the word comes from, confocal. So if I were to watch where that light goes, you can see that the crossover point, that confocal point, is going to allow the light from that one plane of focus to get through, because we have that, that pinhole at one area unit, which is however many microns open. All right, now, here's the trick. If you keep everything right where we are now, what happens with light from the blue dot? Well, it's a little closer to that first lens, so it's going to be coming up at a different angle. And what it's going to actually do is cross over below the pinhole. And then it crosses over and it's going to hit the pinhole on the other way. So it never makes it through the pinhole. So therefore it never makes it to the camera. So we never see it, right? Likewise, if you look at the green one, it would actually focus beyond the pinhole. But before it gets there, it hits the pinhole first. And again, it gets removed from the light path. If the pinhole was a little bit larger and let the light through, it would actually cross over up that way. And so this is how a single pinhole is going to be able to remove any of the light that's out of focus, above or below our plane that we're dealing with. And so if you think about this little diagram for a second, what would happen if that pinhole was larger? If that pinhole was larger, we'd be letting through more light. Hooray! We got a brighter image out of that. But what's going to happen? It's going to be getting that light by getting some of that out of focus light above and below our plane of focus. Boo! We've just lost resolution. So by closing down the pinhole, we get better resolution, which is what we're looking for. Yay. But by closing it down, we also lose a lot of light. Boo! Which is something that we're going to have to deal with. So this is what I was talking about with that balancing act. So the one area unit is where we start, okay? So hopefully that made sense. Now, what does this do for us? Let me show you a couple of slides just to show you the result. If you take a look at this, we are actually taking multiple pictures through a small stack of brain tissue. And each one of this very jerky little movie that you're looking at is a single slice. So we're focusing down through it. Every single slice doesn't show you much because it's only showing you the parts that are in focus. All right, that's great. So if we take this entire stack and we put it all together at the end, you're going to have an image that is made up of nothing but sharp images, no out of focus light at all. And this is how we can get these very sharp images out of confocal. To compare this exact same tissue with a wide field microscope with traditional fluorescence, it's going to look like that. So this is one of the reasons why we come and we use the confocal. So I belabored that a little bit because understanding pinhole really is the heart of confocal. It is how a confocal works. And that concept of larger pinhole, less resolution, smaller pinhole, better resolution is kind of critical to a lot of experiments. All right, I'm going to work my way down over on the left side in the software here and come to the next thing. So we're looking next at zoom. So as I mentioned in the videos, we're going to be working with a 63x objective here, which has a numerical aperture of 1.4, which is pretty high. So we can get some pretty decent resolution out of this system. Now, if we're only capturing it on a camera, if you will, on a detector that's 512 pixels by 512 pixels, well, where's that resolution come from? Because we're giving it lots of resolution, but it's got these massively sized pixels and you're not going to see it. So what we do is we change the format size, make it out of more pixels to work with that. Or the alternative is to zoom in more, make your subject bigger so that it covers, you have more of your specimen covering those pixels. So it's a combination really of that format size we talked about before and the zoom, which is going to give you total resolution. So with the zoom right here, I only put it at whole numbers for some reasons that we might be able to get to later, but just take it for granted right now. That's what we're doing. So we're going to do 1x, 2x, 3x, things like that. 1x means no zooming at all. 2x would be obviously 2 times, 3x would be 3 times, etc. So we can get that way by moving the slider, which I don't really want to do, or I can just type in where it says 1 right now, I can type in 3 and we're at 3x. Or I can go back and put it back where I really want it, which is 1x. Okay. Now, these fun little arrows underneath there, these are all about nudging your image around. So if you are at 1x on your image, you're seeing everything. So the only way to get to a new field of view is to use the little salt and pepper shaker that I talked about in the videos and move physically your stage to another area. And that's fine for lower magnifications, but what you'll find is when you're at very high magnifications and you want to move it just a tiny little skosh, you know what's going to happen, right? You're gonna suddenly slip and okay, your cell's gone. You're never going to find that spot again. So what you can do is if you are zoomed in at all, we can use these arrows to nudge the image around. It would be like, here's my whole big area. I got a cutout in the middle of it, and I'm nudging the cutout around within the bigger area. So that's what these big arrows are for, and you'll see those in use later when we have an image. Now, over on the left, there's some numbers that are relatively important, and I'm not sure if you're going to be able to read them, so I'll read them out to you. The first one here says image size, and it says 246 microns by 246 microns. And what that basically means is my field of view. So this entire integrated system knows what magnification you're at. It knows what objectives in there, what zooms are in there, what other magnifying lenses are in the system, puts it into a formula, spits out, and it knows exactly what your field of view is. And that's what that's telling me. So right now, under my current conditions, I am going to see a field of view of 246 microns squared. All right, that's what we're set up for. Okay, that's fine. Now, directly underneath that, it says pixel size. And this is actually one of the more important numbers to deal with. So the pixel size right now says 481.47 nanometers by 481.47 nanometers. That is the size of a single pixel at 512. Now, if I change the format size to 1024, it's going to change the pixel size because now I've got the same area, the same field of view, but now I'm dividing it up over more pixels. So now the pixel size is smaller. It's 240 nanometers by 240 nanometers. All right, so that's one way we can get some more information in there. So why is that number so important? Well, I like to tell my trainees that the magic number in our confocal is 80, 80. And that is the pixel size number that you are aiming for. Anyone who works in microscopy might understand that there's a term that when you increase the magnification, you get to a point that's considered empty magnification. And empty magnification is really the point where as you're getting in closer and closer to a small little fuzzy dot, it gets bigger, but it gives you more information. When you hit empty magnification, you're getting to the point where that small fuzzy dot gets to be a big fuzzy dot, but without any more information in it. All right. And with this system, that point is about 80 nanometers. Now, this is a rule of thumb. It's not completely true all the time, but it's a good thing to work with on a day-to-day basis. When we have people that are doing extremely specific experiments, we get into this in a lot more detail. And I have a little app on my phone that lets me figure out exactly the correct pixel size. Often it's down closer to 42 or 57 nanometers, something like that. But 80 is always a good number to work with. So as we adjust our zoom and as we adjust our format, we can change that pixel size. All right, underneath that, there's another number. It says section thickness, 0.772 microns. The section thickness, if you think about that diagram I showed you before, it's how thick, or how thin really, is my slice, right, that I'm taking through my cell. And the only way to control the thickness of that slice is, we already talked about it, it's the pinhole. If I make the pinhole bigger, then I'm going to make my section thickness thicker. And so I'm going to see more out of focus light above and below that plane of focus, right? If I make my pinhole smaller, then I'm going to make my section thickness thinner. So I get more resolution in the Z direction as well. So good stuff all around with that pinhole to get more resolution out of it. But remember, if I make it smaller, I lose light, and that's a problem. Anyway, this is going to be an important number when we get to 3D stuff next month. So for right now, we're going to go past it. Okay, next we come to a bunch of stuff here called averaging and accumulation. So we have basically four combinations. We have a line average and a line accumulation, and then we have a frame average and a frame accumulation. So the first question is, what's the difference between a line and a frame? So when we're scanning, you're going to be scanning by line. It scans across the line and it scans down. It's a raster scan. So for those of you who are familiar with a raster scan or a TV screen, that's the way it works. Scans across one line at a time. And so we can average that line, or we can accumulate that line before we go to the next line. So that would be doing these things by line. The other way is to do it by frame. And frame is your entire image once you have that image up there. So you scan the whole thing once, you see an image, then you scan it again, and you combine those two images. So that's the difference between a line and a frame. Now, why are we doing this combination in the first place? What am I talking about? For that, I am actually going to go away from here, and we're going to go back to my PowerPoint. So what we have here diagrammatically are two pixels side by side, right? Or at least very close together. And when we scan, we're going to scan once, and we're going to go right through, and both of those pixels are going to fire. And to give us a round number, let's just say they give us an intensity value of 100. So both pixels are now part of my image, right? Now they're both giving me a value of 100. A value of 0 would be like black. There'd be nothing there, right? So both of these are part of my image. But let's scan this a second time and see what happens. If I scan it a second time, oh my goodness, that first pixel fired, but now the second pixel didn't. So what does that mean? Well, what it means is that second pixel is actually not part of my real signal. It's actually noise. It's a transient signal. So it will fire because of random electronics in the system or a whole variety of things that would be a whole lecture in itself. But the bottom line is we have the difference between pixel 1 is real signal and pixel 2 is noise. And so what we want to do is deal with this signal to noise ratio. We want to be able to get more signal and less noise. And the way we do that is by doing this averaging. So let me show you how that works. If we've scanned twice, right, we get this. Let's scan it a third time. Oh, good. You know, we're still getting it that first time. We're getting the signal in that first one and nothing in the second. Let's scan it a fourth time. Oh, darn that pesky little noise. It came up again. And then the fifth and then the sixth. All right, so if we take these numbers and we average, so we got 6 scans, 100 each time, divide by 6. All right, you got 100 average, right? That makes sense. So our first pixel, the intensity of that in our image is going to be the equivalent of 100. Great. Well, if we average the noise column over on the right, you're going to get 33.3 instead. And that's going to give us then a 1 to 1/3, basically, signal to noise ratio. And this happens because of the averaging. So instead of just taking the image once and being done, we take it once, we scan it again, we scan it again, we scan it again, and each scan, the image looks better and better and better. Less noisy, less noisy, less noisy. You do reach a point of diminishing returns, and so for our talk today, we're only going to do 6 averages. All right, now, how is averaging then different from accumulation? Because remember, we have those 2 options. Well, remember I was telling you before that we had a problem with the amount of light coming into the system. For instance, if we wanted to close down that pinhole to get more resolution, we're cutting out a lot of light. So where do we get more light to make up for that light that we lost? One way could be accumulation. So if we scan 6 times here with accumulation and we get essentially the same results, how does this work with the signal to noise? Well, with accumulating, you're adding everything up instead of averaging. So our first column is going to be, instead of 100, it's going to be 600, which is great, right? A lot brighter. Well, our noise is also going to be brighter. It's going to be 200, but guess what? 600 to 200 gives us the same ratio. So by accumulating, you're still getting that reduction in the noise. You're still getting better signal to noise ratio, but the advantage here is that your signal is getting brighter and brighter and brighter as you do it. So if you've lost light by closing down the pinhole, as a for instance, you can gain it back by doing accumulation. And all it costs you is time. But that can be a problem too. All right, let's get out of here, and we are going to get back to our settings. So what we're going to do is we're going to do a frame average, which is the bottom left one of these 4 little black thingies, and we're going to make that 6. So we're going to average the image 6 times when we do this. I generally do not start with any accumulation because of the time that it takes. It is a very useful feature, but it does take time. And so I kind of keep that in my back pocket as a way of being able to make things brighter if all else fails. But if I was going to use it, I would only do it by line. And there's a good reason for that, because as the scan goes down, I want to be able to see it live, or as close to live as possible. So it's going to scan each line however many times I tell it to do it. And so it's going to look as bright as it will as soon as I see it on the screen. If I accumulated by the entire frame, it would scan the entire frame once, and then scan the entire frame a second time and add it to the first one, and then scan it a third time and add it to the other 2. And so with each scan, it would get brighter and brighter and brighter. So I wouldn't actually see how bright it was until I got to the very last scan. So accumulation works better by line, and averaging works better by frame. Okay, underneath those we have rotation. This is just a way of essentially rotating the camera, if you will, because you can't really move the stage around. And if you want your cell to kind of rotate on the screen and fit the frame a little bit better, you can rotate the image around so that it fits better into the frame. So we might want to do that later right here. And by the way, where you see here this rotation, or up here where you saw zoom factor, we see this little slider. We see, oh, sorry, I was pointing at pinhole, which also has the same thing. You see the little slider, you see a little box with numbers, and both of those do exactly the same thing. I can move the rotation with the slider. I can move it by typing in a number. I also have something else that's down here on my table, which I can't quite show you, but there's a whole line of knobs that I'm looking at. And I can adjust those knobs, and those will also change the same thing. So 3 ways to do exactly the same thing. So you'll be seeing me make adjustments, and you won't see what I'm doing, but I'm doing it on the knobs down here. So rotation would be one of those things. All right, we set our pinhole at 1 area unit. We've left it at area unit. Next, we get down to Z-stacks. And you know what? We're not going to talk about Z-stacks today. We're not going to have time. That's why we're doing this workshop in 2 parts. So July is going to be about doing 3D stuff, so you're just going to have to come back and listen again. So I'm going to close up that little box and we're going to continue on. Sequential scan I mentioned before, we are going to use, but we're going to come back to that. So next, we're going to come up over to the right or the middle of the screen really, and we're going to look at our lasers. So remember in the videos, we talked about turning on the laser power, turning on the key. We started this process by going up to configuration and turning on our lasers there. They're still not on yet because we actually haven't set some other parameters here. So there's a couple buttons up here that we have to check. So we have two little red dots up in the top middle here. These are now active, so the lasers cannot be actually on unless those are active. And you also notice that we have a whole bunch, we have 9 lines that are represented here, going from the 405 laser on the left, and then over to the right, we have in order 458, 476, 488, 496, 514, and 561. Now the 458 up to the 514 are the 5 different lines in the argon laser by itself. So the argon laser is responsible for all 5 of those lines. The 561 is a laser by itself, and the 405 is a laser by itself. But these are the lines that I have available to me. They're all set at 0%, as you can see at the top of the little bar. So what I have to do is give them a number. So if I go to the 405, which is my ultraviolet, it's not really ultraviolet, but we call it that. For the Dappy, it's at 0%, and I'll tell you that our starting point is usually 7% on this, and that's going to now, at this point, that's really going to turn on the laser, and it's now really available to us. You see that there were some cool little orange lines that showed up, which indicate that it is now working, and in the spectrum in the middle of the screen, you see a 405 and a vertical line showing us on that spectrum where 405 nanometers actually is. So this is now showing me where the 405 laser is and that it's turned on. All right, now I mentioned before that we are using Alexa 488 as one of our dyes. So I just wanted to also point out, some of us are old enough to remember some other older dyes, and maybe traditionally you might have learned some of these fluorescent dyes in the ancient past. And one of the most common green dyes that everybody always used was FBCA, or Fluorescein, basically. Well, also those of you who have worked with ophthalmics and stuff know that Fluorescein is also used as the dye for fluorescein angiography, among other things. And if you think about how it's used in there, again, forgive me for those of you who actually do this. I'm kind of simplifying things. But the way that fluorescein is used in a person is it's injected into, I believe they're carotid. The dye finds its way up to the back of your retina, where you can take an image of that from the front of the eye, and that's how you can get your fluorescent picture of your retina. So forgive me, Howard, if I'm wrong on that, but it's pretty close. Anyway, my point is that you now have an eye that's got this fluorescein in it, which is light sensitive, and I'm sure that doesn't feel very good, does it? So it's very nice to know that fluorescein is going to fade away and kind of work its way through the bloodstream and be gone. The fact that it fades away is a very useful thing in this case. Now, what did I just say? I just said that this is a dye that's designed to fade. And didn't I just tell you earlier that when we are imaging, one of our biggest problems is fading? So why would we use a dye that's designed to fade when we're doing imaging projects with a confocal, especially when we're using a very bright light? Well, we don't. We try to avoid FBCA as much as possible. Instead, there are newer dyes out there that have been engineered to withstand the intensities of the lasers. And most of these dyes come with a number. So Alexa 488 is telling us that we should use a 488 laser, nanometer laser, to excite that particular dye. And that's going to replace something like FBCA. So that would give us our green emission. So I'm going to be using the 488 laser line here. So right now, I'm actually going to be setting that at 4% on the laser. I'm not going to get into how I got to these numbers. We're going to run out of time if I do that. But this is usually where I start. Okay, and you can see down here that the 488 laser line is now showing up on our spectrum. All right, the 561 is the last one, and we're going to set that at 2%. Now, why are these all different percentages? Well, because all of the lasers are different powers to start with. And what we want to do is use a certain percentage of each one of those lasers to make sure we don't really get above 1%, or excuse me, 1 milliwatt on each of the lasers, so we avoid burning out our cells and we don't create too much of that fading and quenching that I was talking about. So all of these lasers then replace our excitation filters on our traditional filter blocks that we would use for fluorescence. I'm not going to talk about dichroic so much because this is a magic confocal. It uses what's called an AOBS, an acoustico-optical beam splitter. And the AOBS is essentially a universal dichroic. It's a big crystal. You put some electricity in one end and you can tune it to a very specific wavelength, and that's going to pass certain wavelengths and reflect others just by that positioning. So the AOBS can be used for basically any of these wavelengths across the system. And so that basically makes this system very spectral. It's a spectral imaging system. All right, so if you think again about that filter cube that we talked about, we have our excitation filter taken care of by the lasers. We have our dichroic taken care of with the AOBS. And now what about the emission filter? Well, we don't need an emission filter because what we're going to do is we're going to take multiple little cameras or multiple detectors, and we're going to set them to look at only specific wavelengths. So it's kind of the same thing that a filter does. A filter only lets through certain wavelengths. We're going to tell the detector here, only look at these wavelengths. So it's kind of the same thing. So if we look at the software here, you can see underneath the spectrum, we've got this line of 5 different little rectangles with little arrows on them. These represent my 5 different detectors. We have 2 different kinds of detectors on this confocal. We have PMTs, which are the traditional photo multiplier tube. We also have HIDs, which are hybrid detectors. The hybrid detectors are higher resolution. But, excuse me, they're not higher resolution. They're more sensitive. There we go. They're higher sensitive. They have higher sensitivity. So you're going to be able to see a dimmer light source is going to, you'll be able to pull that out. The downside, that's what I was trying to get at with all my stumbling. Forgive me on that. The downside to these detectors is, yay, they are going to give you more for your buck, but they're also going to do it with a much lower resolution in the tonal range. So they're a higher contrast detector, which is great if you have a low contrast original, but if you've got a original that's got high contrast to begin with, you're basically going to wind up with black and white instead of a range of grays. Dappy is one of those dyes that has everything from very dark areas to very light areas. And if I try to look at Dappy using one of these higher sensitivity detectors, these higher contrast detectors, what it's going to do is going to take all the dark areas and turn them black, and take all the brightest areas and turn them white. And then it's going to make everything else in between a little bit too much, too dark, and a little bit too light. It's basically going to make it look like Swiss cheese, which means we're going to lose that finer tonal information in our image. So for something like Dappy, I try not to use the hybrid detectors. I'm going to use a PMT for that. But most of the time, things that are very, very weak, we're going to use the high-D detectors for. All right, so that happens to be very useful in our case because the first detector in line here happens to be a PMT detector. So we're going to start by setting up this PMT detector over on the left. This is PMT1. So to begin with, I can adjust this slider. This slider represents my detector. I can make it smaller, I can make it bigger, I can move it around, but you notice I can't move it past this other one over here. I bring it up to the right, and it just hits and stops right there. What I'm going to do is double click on this, and it's going to give me the option of putting in the range. So this would be just like saying, okay, detector, I only want you to look at, and I'm going to set it at 415 nanometers, which is 415 nanometers, which is 10 nanometers away from my closest laser line on the left. And it's going to go up to we're using a 488 laser, so it's going to go up to 478 nanometers on the other side. So now what you see is this detector right here, which is showing up in red, is 10 nanometers away from the closest detector, the closest laser on either side. This is important because detectors are pretty much like any other detector, like your eye, for instance. Your eye is a light detector, and your eye isn't supposed to look directly at a laser light source, and neither are my detectors. You can burn out your detectors. The HIDs, in fact, if you do park them right underneath one of the lasers, will automatically turn off to save themselves. So you got to be careful about where you set these detectors up. So right now I've set it up between those two laser lines. All right, now I haven't actually turned it on yet, so I'm going to come down here where it says active and turn it on. It is now active, and this gray bar shows up in between, showing we're just connecting to that. We have nothing selected here for a dye. It doesn't really matter if we have anything or not, but I am going to come in here because I'm going to select Dappy, which is right here, as the dye that we're using for this, because by choosing that, it puts, this is going to be hard for most of you to see, but there is a very thin white line that's going along the spectrum here, which showing us the emission curve for Dappy. Now, the reason this is important is take a look at this emission curve. It hits a peak somewhere between these two lasers, but it keeps going way, way up into the 600 range, which means that when the Dappy emits its light, it's going to be emitting all the way through, all the way up into the 650 range, which is also where the mitochondria are going to be emitting, where the actin is going to be emitting, and so we're going to have problems with overlapping signals because of that. I'll show you that. All right, so that curve helps us try to figure out what we can and we can't do. All right, now you notice also PMT1 has a little green bar next to it, so we can select that. These detectors are monochrome detectors. They're black and white. They are going to only look at, is there light and how bright is it? They're not going to say, oh, I'm between this wavelength and that wavelength, so that's this color, so I'm going to show it to you in blue. It only sees it essentially in black and white, and so you can give it a color. So I can give it blue, which I will right there, just for tradition's sake. But the reality is, in this day of understanding that there is a good percentage of the scientific population that is red-green colorblind, we might want to actually change that from blue to perhaps yellow. We would be using cyan, magenta, and yellow instead of the traditional red, green, and blue. So maybe I'll do that one instead. Okay, so we have it set up for yellow. We have it for Dappy. We're basically ready to go, except one little thing that we forgot. If I click on PMT1, you're going to see down here that my gain setting is at 0, which means even though the camera, if you will, the detector is on, it's set to a value of 0, which means it's not on. So what I need to do is bring it up to a value that we can actually work with. Now, the lower the gain level, the better the noise. The higher the gain level, the worse the noise gets. So what's the best point? Somewhere in the middle of the range. So for this system, it's somewhere around 650 nanometers. So that's about where I start. Or not nanometers, 650. I'm not even sure what the units are. So we're going to start with it at 650 units for our gain down. All right, and that's going to fade away. Underneath there was also an offset. That's our black level. We're going to keep that in our back pocket for later. All right, now let's real quick set up the other parts of the system and the other detectors. So the next detector in line is one of those high D detectors. I'm going to turn it on. We're going to go with standard mode instead of bright R or photon counting, which are other options. We're going to make this Alexa 488 so we can see the curve. And here you can see what I was talking about. If this is the curve for Alexa 488, and this is the curve for Dappy, they're completely overlapping each other. So if I put this detector underneath, that detector is not going to know if the light is coming from the 488 or from the Dappy. It's going to show you both. Now I'm going to double click on that, and we're going to set it again at 10 nanometers away from both of them. So we're going to go at 498 on the left and 551 on the right. All right, so we're kind of all set there. Now the high D detectors by default start off with a gain of 100. So we're actually going to leave it there. For the high D detectors, we actually don't want them to go any higher than 100. As with the PMT, the lower the gain, the less noise. So less than 100 is great. But higher than 100, all it does is it gives us more noise, but it's kind of a law of diminishing return. It'll make your signal brighter, but with so much noise that it's not worth it. Okay. All right, our next detector is the PMT3. We're not using this one. I'm going to turn that one off, and I'm going to turn it down into a little tiny sliver, and I'm going to move it right up against high D2 so it's not parked right underneath the laser. Even if these detectors are turned off, they still can see the laser, so you don't want to park them right underneath. All right, I'm now going to turn on high D4, and it basically works the same way as the last one. I need to try to find MitoTracker red here. That's the curve for MitoTracker red that just showed up in the spectrum. Now, in this case, I'm actually going to do something. There was a workshop I went to on Confocal once. So if I start here and I set my lower range for this detector at 571, which is 10 nanometers away from the laser line, where do I set my other end? Well, this workshop was given by a guy out in Pittsburgh, and he had this saying that sticks with me to this day. He said, every photon is sacred. I love that one. The reason he said it is because in any of these fluorescence systems, especially confocal, one of the other big problems is loss of light. And so the efficiency of the system is paramount. So if you're losing any of the light anywhere in the system, you want to optimize your whole system so that you collect every single photon that you can. Well, if we know that the MitoTracker red is going to be emitting way, way up here, let's just take this detector and bring it all the way up to the end. Let's get every single photon that might be coming out of that, because every one is sacred. And that way we get as much light out of that as possible. Okay, so there we go. And high D4 is also set up for 100. And we're just about, ready to go on that. 1 hour and 8 minutes right now. So about an hour after I started this. And it took that long to get to the point where we're about to see a picture. Okay, so the first thing I'm going to do is come back over to the microscope. And if you remember all that fun stuff from the video, I'm going to pick a tooth, in this case, RFP, or the red one for the MitoTracker. I'm going to punch him in the nose, and I'm going to look in and make sure that my cell is still in focus, because I put it in here a while ago. I'm going to use my salt and pepper shaker to focus on that. And there it is. It's at least close enough to what I can see by eye. And then I'm going to punch him in the nose to turn that light off. Again, as I said in the video, that light is not the laser light. That's the light coming from the traditional fluorescence light source that's sitting on the table here. Now what we're going to do is we're going to come down to the bottom of the screen, and you can see over on the left we have a live button next to a, there's one that says auto something again. Remember, stay away from that one. Why they put it so close to the live button, I'll never know. But I'm going to hit live, and you are now going to start to see the image coming up on the screen. Now, I'm going to stop right there, because one of the things that I always try to tell people is for every scan that you make, you are quenching your sample, right? You scan it once, scan it twice, you scan it, you know, every scan is going to do it. So the best way to work with all this stuff is to scan it once and stop it. And then look at the screen, analyze what you're looking at, figure out what to change, make some changes, and then scan it again. So right now I'm going to do that. And one of the things that I need to do is move all you guys out of the way. So I think you probably see see you on the screen. I'm going to put you down in the middle here. That's an area that's not being used. So over on the right-hand side of the screen, what am I looking at? What I'm looking at in the top left is my Dappy signal. On the right, I'm looking at my actin signal from the 488. And on the bottom left, I'm looking at my MitoTracker red. Well, some of you might have been saying, oh, but he didn't change this and he didn't change that. And you would have been right. If you were here live with me, you could have said something, but you weren't, so I had to figure it out for myself. One of the things I forgot was on high D2, that's still set to red. So that's why my actin filament is showing up in red. Well, I told you before, instead of using red, green, blue, we're going to use cyan, magenta, and yellow. So I'm going to actually set the actin filaments to cyan. And then I'm going to click down here, I'm going to go to the high D4 detector, which right now is just set to black and white. So if you look down here in the bottom right, you can see the mitochondria showing up in black and white, really. That's because my detector was set for black and white. So I'm going to make this magenta. So now if I hit live again, now we have the right three colors that are showing up over on the right. Okay, now, so this is channel 1, Dappy, channel 2, which is our 488, channel 3, which is our MitoTracker. In the top right here, you can see channel 1, channel 2, and channel 3, and underneath that, an overlay button. So I'm going to click on that, and here we can see in the lower right the overlay. When I'm on the screen, if I click on the actin filament one, you can see this white box is now selected around that. If I go to the mitochondria, the white box is around that. So whichever one of these is selected is the one I'm working on at any given time. If I double click on that box, it makes it bigger. If I double click again, I go back to the whole thing. So this is how I kind of navigate around and look what I'm looking at. All right, now let's think about color for just a second again, and think about the fact that our eyes don't see color the same way. All of our eyes look at color a little bit differently. And different colors themselves are also going to be seen differently with the same eye. So the reality is the best way to look at this objectively is to use an under-over lookup table. So I'm going up to the top middle here, and I'm going to click on this button, which is going to give me an over-under lookup table for our images. So this is the way I like to work with everything. The Dappy is showing me basically lots of different versions of like an orangey red color, and that's it. The other two are showing me green along with some orangey red color. So what does all that mean? The green is telling me that any pixel that's green is black. It's at a value of 0 in that pixel. If that pixel is fully saturated, then I would be seeing blue pixels. And that's how I tell that I'm overexposed. So by leaving it objectively like this, I can tell whether I'm overexposed or not. And this, again, is critical for doing any kind of quantitation. So when I look at all this and I look at the Dappy, what I realize is there are no black pixels in the Dappy, which means my darkest pixels are still some level of color. And my brightest pixels are still some level of color. So what that translates out to is this is going to be kind of muddy when we get there. And we're going to work with that a little bit later. Now, why do these have the green in them? Well, they have the green in them because remember I said these two are high D detectors. So the actin filament and the MitoTracker down in the lower left, we're using the high D detectors. Those are higher contrast detectors. They're taking the darkest pixels and turning them black. And so we got green pixels. Now, overall, it's not quite bright enough yet to see any oversaturated pixels. So this gives us something to start with. Now, what we're seeing on the screen, however, is a little hard for you guys to see, I think. So I'm going to zoom in a little bit just so we can see some more detail. I'm going to go in 3 times on the zoom. So if I come over to my zoom factor and I adjust that to 3, and I go back to my live view, that's going to zoom in now on my sample a little bit. And if you notice down in the MitoTracker here, oh, there are some blue pixels in there if I make it bigger, we can start to see that I do indeed have a little bit of oversaturation in the mitochondria. If I look at the DNA from the Dappy, I do not. And if I look at the actin, I do not. Well, I have some in the middle of the big round circle, which we're going to get to in a minute, but not out here where my actin filaments are. All right, so remember I also told you that when we are zoomed in, so I'm going to go live again, I can now use these little arrows over on the left in that little black square and kind of nudge that image around a little and put it down in the middle of the frame. So that's how I would use those. And then I'm going to be focusing up and down through my sample. So right now I'm going from the top to the bottom in both directions, focusing or slicing, if you will, through all parts of my cell. I'm going to stop right here because this is kind of the brightest part of it. I'm going to stop right there. So again, if I go back to the mitochondria now, you can really see how bright that is. That's too bright. My biggest question, though, is what the heck is that? So what I'm pointing at is the big round blob in the middle of the actin filaments in the top right corner in my high D2 detector. So if you think about what I was talking about originally, about overlapping signals, we can kind of explain this. Right now, I am only doing a scan of everything at the same time. So we're doing this simultaneously. In the scan, it's doing all three lines, all three channels at the same time. And so when I hit live, the Dappy, the 405 laser is exciting, which means the Dappy is also exciting and emitting. And so the Dappy is emitting, and at the same time, the 488 or the high D2 detector is detecting. And so guess what? That's what's happening up here with the actin filaments. I'm getting not only these very weak actin filaments, which you can kind of see in the background very weakly, but I'm also getting the bright nucleus because it's bleeding through. So the term bleed through comes from this, and we realize right now we cannot do these simultaneously. So what I'm going to do first is come over to the left, and we're going to change our sequence. So instead of having a single scan, we're going to do a sequence of two now. I'm going to click on the plus button. I'm over on the left-hand side of the screen now, and I now have two scans. Now, I haven't changed anything else, so both scans are everything twice. So if I hit live right now, I'm actually going to see 7 boxes. I'm going to see the Dappy twice, the actin filament twice, the mitotracker twice, and then the overlay once, because it's overlaying everything that I'm doing. So what I'm going to do is I'm going to click on scan 1, and I'm going to say, all right, in scan 1, I just want the Dappy. So I'm going to turn off the other 2 detectors here and here, and I'm going to take the lasers for both of those, and the 488 and bring it down to 0, and the 561 and bring it down to 0. So now when you look up here, for scan 1, the 405 laser is on, and the PMT detector is on, but that's it. The other stuff is off, right? Everything stays where it is, but those are just turned off. Now I'm going to go to scan 2, and in scan 2, remember, everything was on, but in scan 2, I want to do the 488 and the 561 together. Because if you look at these 2 scans, or you look at these 2 curves for the 488 and the mitotracker red, you can see that they are pretty well separated. So any overlapping signal in this area in the middle, we might be able to handle. So I'm not going to worry too much about that right now. But I do want to turn off the 405. So I'm going to take the 405 laser and bring it down to 0. And I'm going to take PMT1 and turn it off. So what I just did was I changed from a simultaneous scan to a sequence. And now when I hit live, you're going to see the difference. Ready? Here we go. Click on live in 3, 2, 1. Oh, goodness, look at that. My nucleus is now not showing up in the 488 signal. It's not supposed to, because it's not exciting and it's not emitting now when I look at that channel. So this is going to be a lot more realistic, and we're taking care of some of that bleed through stuff. All right, so that's a good place to start. All right, now let's take a look at Is this overall a good exposure? Well, if we start with looking at things, we can see that our Dappy is actually a pretty decent exposure. It's a little bit too bright. I can make it dimmer by turning down the gain. And that's basically what I'm going to do. But I got a big problem here with the actin filaments. They are not that bright. If I change back to like a black and white view of that, you can barely see them. And if I change it to the color view, you probably can't see them at all. So if I go back to the over-under lookup table, this is another reason for doing it this way. You can see that there's something there. It's just a little bit beyond your visual acumen there. So we need more light out of this. So let's decide, how do we make the 488 signal brighter? Well, let's see. I could turn up the gain some more, right? Well, let's take a look. My gain for that detector by default was 100. And I already told you that with the high D detectors, we don't want it higher than 100. So that's off the table. We're not going to turn up the gain. All right. We could open up the pinhole. We talked about that, right? Well, if we open up the pinhole, we have to open up for all of them. They'll all get brighter. And we'll also lose that resolution we were talking about. So that's not a good option for this one either. All right. Let's forget that one. What else can we do? Well, we could do that accumulation I was talking about. Well, again, we could do the accumulation, but we, again, would do it to all of them, and it would take a lot more time. So it's not a bad option, but maybe there's another one here. Okay, how about turning up the laser? Well, let's think about it. This is Alexa 488. This is not FBCA, right? This is not that eye-popping, one that I talked about before, that dye that fades on. Alexa 488 is a dye designed to be used with lasers, so it's brighter and more stable under a laser line. So if I'm using an Alexa dye, I feel fairly confident that, you know what, I can turn that laser up a bit. So I'm going to do that. I'm going to go from 4%, and I'm going to actually bring it up to 10%, because I know I can do that. Some of this is experience. Some of it is some other, formulas that I worked out in advance. But 10% is going to work. Now, if that's the only thing I change, then the actin filament should get brighter. I'm going to go live once and see what it did. Ooh, much better. Look at that. Now I see something. Okay, I like that. Now, however, you know what? I think I'm going to go up to 12. I think I can go a little bit higher on this one. Ah, there we go. I'm starting to see some blue pixels up along the top. So I'm reaching the maximum that I can do for that anyway. So that's going to give me a decent range there. All right, now let's take a look at the nucleus over on the left here. No black in the background. The nucleus itself is a little bit on the bright side, or a little on the dim side, because there's no blue pixels. So what do I want to do? Do I want to increase the laser on this one? All right, well, the problem with Dappy is it does fade a little quickly. So I probably don't want to turn the laser up anymore. So how am I going to get this one brighter? Well, I could turn up the gain. Let's see what happens if I do that. Right now I'm at a gain of 650. On my knobs down here that you can't see, I'm going to go up to 800. And I'm going to go to live. Oh, good. That made it a lot brighter, but it also made it a lot grainier, a lot noisier. And I'm going to adjust an offset, which is my black level, down a little bit, so that now when I look at that, I'm going to see some black. And I'm going to bring down the overall brightness a bit. So now I'm down around 730, 722. All right. All right, that works for me, actually. I'll go with that. When it comes to the gain settings, like I said, it's all a compromise. The lower the gain, the less noisy the image. That's great. But, sometimes it's just not practical. I'd like to keep it down in the 600s, but sometimes I have to go higher. How high will I go before it goes too high? Well, 750 to 800 is about my max, and I'm still a little less than that, 723. It does look noisy on the screen, but remember what you're seeing on the screen is a single scan only, no averaging, and you're seeing it at 512 by 512 because we're live. So overall, this image on the screen doesn't look that great yet. All right, So what's next? Let's take a look at our mitochondria. Our mitochondria are too bright. I'm going to make them bigger so you can see what I mean. Definitely too bright. So I'm going to just take this one, and with a gain of 100, I'm going to turn it way down to around 40 or so. See what that looks like. Go live. Okay, that's better. I'm going to turn it up a little bit, maybe to about there. Okay. I do see some blue pixels, but not too many. So I'm around 45 right now. All right, now, what I want to point out here, though, is what's all this in the background that look like actin filaments? Well, guess what? They're actin filaments. Those are bleed through from this tiny little bit of overlap. If you look on the screen in the middle, where we have the two curves for the 488 and the mitotracker, there is an area of overlap in between. So what I can do is I can move the detector to get it a little bit away from that overlapping area to try to minimize that a little bit. But at the same time, this really isn't that bad. If I change the black and white view, you really are going to have a hard time seeing them. If I change the color, you're really going to have an even harder time seeing them. So I really probably don't need to adjust it too much, but I will just for sake of argument. I'm going to take the detector and I'm going to eyeball it. I'm going to bring it up to about there. Now, what I want to do is I want to make sure that the detector is still seeing the peak of the emission, but I'm trying to get it away from the worst of the overlap. And so if I do that and I go live, okay, they've been reduced a bit more. And you know what? I'm going to live with that. So it's all about trying to make these compromises and deciding what works for you and your experiment. You need to understand both your options as well as your experiment to be able to do that. That's why the training takes as long as it does. A lot of what I'm doing here is trying to explain why you're making the choices. You notice I'm not saying click on this button and change it to 600 and not explaining why, because that's pretty much the same thing as going auto. You know, it's like, okay, click on here, make it 600 every time. Well, it's not going to be 600 every time. It's going to change. And you need to understand when to make those changes, when to adjust that balance to make it right for you. All right, so I'm back to here again. Now we're looking pretty good. Okay, so I have my image, or at least the first start of it. And I'm looking at it and say, all right, this is fine. I am now going to switch my format because I'm ready to take this picture now. So I'm going to come up to the format in the top left corner here. I'm going to get away from that 512. I'm going to go to 1024 by 1024. As I said in the beginning, that is usually how we do it. So I'm going to go live one more time just to take a look at it. You notice the scan seems a little slower now. It is. It's going through twice as many lines now. And it also looks like it's a better resolution now because it's made up of more pixels. All right, this looks pretty good. Okay, so what I want to do now is I'm going to actually take the picture. But what I'm going to do is I'm going to, again, zoom in on the mitochondria. Well, probably not the mitochondria. Let's do it with the actin. This is better. So you look at the actin filaments, and you see that this is a pretty noisy image with one scan, right? Very, very speckly, very noisy. So a single scan is not going to do it. And here's where I want to show you how that averaging works. So when I hit the start button at the bottom, that's going to start the imaging process. And it's going to start any sequence that I've got going and anything else. So we always hit start when we are ready to take the picture. I'm going to hit start, and I want you to watch the image build over on the right. It's going to scan once, and then twice, and then 3 times. I have it over here set to a frame average of 6. So it's going to scan it 6 times. What I want you to notice is there's going to be a huge difference between 1 and 2, and even 3. A fairly decent difference up to 4. But once you get up to 5 and 6, yes, there's a difference, but not that much. All right? So, we ready? Everybody open your eyes, and here we go. I'm going to hit start. 1, 2, 3. All right, so scan 1 and 2, much better. 3, pretty good. 4, okay. 5, 6, Okay, so not a lot of additional stuff when we got to the 5 and 6. So the reason I say that is because most of the time, for most images, I'm going to scan them with an average of 4 rather than 6. But to get this, if I know I want to get a nice, really smooth cover image, I'll do it 6 times or maybe even 10 sometimes, just to get more and more of that averaging, even though I know it's not necessarily doing a heck of a lot. All right, so how does our overall image look? Hey, not bad down there. Looks pretty good. This is the raw image right out of the gate. Now, when this image is captured, there are 3 channels. The overlay is a combination of those 3 channels. The only thing that we have as raw data right now are those 3 individual monochrome channels. The way they were captured is part of the metadata. So if we're going to do any quantitative analysis on this stuff, it's going to be done on these images basically the way you see them now, but in the background. Now, the reason I say all that is because I'm going to change this to a nice color view instead. And now I'm going to make some adjustments to make a prettier looking picture. So over here on the left, you see a nice tall vertical yellow line now, which corresponds to the Dappy. I'm going to make it a bit brighter by bringing down the top end. And I'm going to bring up the bottom a little bit to make get rid of any black or any little bit of color in the background. I think that looks pretty good. I am blocking it up a little bit too much in the middle, but for now I'll accept that. All right, now I'll come over to the 488 signal, the actin filaments. I'll do the same thing. I'm going to bring that down. The black looks pretty good, so I'm not going to change that. And now I'm going to do the same thing with the mitochondria. I'm going to bring that down to where it looks good. So all I'm doing here is making it look good. So if I look at that, just the Dappy, it looks like that. If I look at the actin filaments, it looks like that. If I look at the mitochondria, it looks like that. And if I look at the overlay, it looks like that. Ugh. I don't like the Dappy. Bring that back up again. Okay. I haven't changed the raw data at all. The raw data is untouched underneath this. Everything you see on the screen is an overlay. And so if I want to get some quick images, some TIFF files out of this that I want to use as part of a figure or something like that, what I'm going to do is I'm going to save these images and export them as TIFF files. But even after I do that, I still have the raw files underneath that I do all my analysis with. So I'm going to come all the way over to the left to the Experiments tab. This is Series 014, which tells me nothing. So I'm going to right-click and I'm going to rename it as 0163X3X, which tells me my magnification. And then I'm going to come back over to the right and I'm going to take a few snapshots, which are literally screenshots. So if I could do a right-click snapshot all, it essentially takes exactly what you see on the screen here. All 3 channels plus the overlay. If you want to go back to the boss and show them one picture, there you go. That has everything in it. But if I want to do a figure, I'm going to probably want to line up some of these things side by side with some of the original data. The original data is 1024 by 1024 pixels. We know that because that's what we set it up for. How big is this snapshot on the screen? Well, the way it's set up right now, if I double click on the Dappy, it fills the space you've got. So it's bigger than 1024. So what I'm going to do is click on, in the middle here, there's a 1-to-1 button. I'll click on that. And you see there's not a big difference, but there's a difference. So make sure I'm at 1-to-1, and I'm going to right-click snapshot, and that's my Dappy snapshot. Then I'm going to double-click on that and go over to the 488 signal. I'm going to right-click and snapshot on that one. Then I'm going to do the same. Oh, shoot. My finger slipped. Do the same for the MitoTracker. And one more time for the overlay. Now I'm going to do one more thing for the overlay. I'm going to put scale bar on. So I can click on the scale bar right here. And what you learn about all the softwares out there is they are horrible. Some are good, some are bad. This particular one allows me to make a scale bar, which is only good as a reference line. Because if you try to use this scale, if you try to use this bar in a figure, it's too skinny, and I can't make it thicker. So what I do is I make a really long line and try to make it straight. And because I have everything at an angle here, I didn't make it straight. But it's about 80 microns across. And the main reason that I wanted to show you that is that. How big do you make a scale bar in a figure? Well, generally it's about a quarter of the image. So 80 microns to start, cut that in half is 40, cut that in half again is 20. So I want to make this bar 20 microns. So if I come up here and I click on my arrow and I right click down here and go to properties, I can change this to 20 microns, hit OK, and now I have a 20 micron bar instead. And I can put that wherever I want to put it. Usually you put it in the lower right. Now again, this is a good reference. You can open this up in Photoshop and put it next to the image that doesn't have one and know how big to make the bar, but I would not use this particular snapshot in a figure. So I'm going to right-click and snapshot. And now if I come back over to the left, all the way to the left, you can see this white box is my raw data. Though that is my original stuff. So I'm seeing everything. I'm going to undo the one-to-one so you see everything the way it was. You notice, by the way, that wherever I move that scale bar, it's on all of them. Excuse me just a second. Okay. We're very close to the end, so I'm not too far off on what I was aiming for. And then if I click on that first one, there you go. That's that first snapshot. That's the one showing all 3 channels plus the overlay. This next one is just the Dappy one. So I'm going to rename this file so I know that for the future. And this is, again, I'm not going to get into all this with you right now, but this is something that I worked out many, many moons ago to figure out how to name my files. This comes in very handy when you look at all your file names without trying to find snapshots or thumbnails by labeling the files in a way that you know exactly what it is without opening it. So if I know that that last one was Dappy, I know that's the Dappy image. This one I'm going to call A488 because I know that's the 488 one, etc. So I am, oh no, again I slipped. Sorry, it's 488. It's this one. Rename. Why did they put the delete right next to rename? I don't know. This is going to be Mito R. By the way, Mito Tracker comes in green, red, far red, different wavelengths. This is going to be Snap OV for overlay. And the last one is going to be Snap OV scale. So I know that's the one that's got the scale bar. Right. So we have now taken all of our images, but we haven't saved anything yet. We still haven't saved anything. We have named it, we've taken them, but we haven't saved them. So I need to come up here to the experiment and give this thing a name. So I'm going to right click and never, 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 I'm speaking from experience, never, rename the experiment. The only time you want to name the experiment is when you save it. So we're going to save the experiment as, and it's going to bring me to the last place that somebody saved something, which hopefully was not in the temp drive. Oh, that's right. Our IT people were up here this week, so we were doing something else. What I'm actually doing is saving to the data drive that's on the computer in this room. We have all of our microscopes hooked up to a central server at Wistar, and we're connected to that from all of the different microscopes, all the different computers. But what we find with the very large files that we create with some of these, that they can get corrupted during the saving process, especially across a network. So we only save directly to the hard drive here, and then we're going to then copy them over to the server when we're done. So for the moment, at least, I'm going to come to here. This is June. I'm going to make a new folder called BCA. So I know where I came to. And I'm going to make a new folder for today called 2021. Today is June 23, right? All right. So for those of you, Hussein, if you are still on from South Africa, I don't know how you put your numbers together. A lot of the world gives the date, like if it was, say, April 7th, that would be 4/7/21 in my lingo, but that might be 7/4/21 in your lingo. So to avoid that particular problem and make sure that everybody saves things the same way here, so we can find things chronologically, we label them this way. So you start with the year, and then you spell out the month, and then you put in the date. So that's the day we did this. And I'm going to now save my experiment. I'm going to call it something as short as I can, which is actually BEC for bovine endothelial cells. That's what's on that slide, and that's all I need to know. So I hit save, and that's it. So now what I'm going to do, last thing, is I am going to right-click, and I'm going to export as TIFF files everything. I'm going to browse to that folder that we just made. And I'm going to hit OK. And I do not want to overlay anything. All I want to do is send the raw data and hit OK. Just did that. And so if I can show you now, in my data drive, in my images, in 2021, in June, in BCA, we do get a lot of folders. Here you have, as you can see, all the little thumbnails, right? And if I double click on them, oh shoot, yeah, later. This is BEC, so I'm looking at the top of the black square. It says BEC, that's the experiment. This is picture 101. It was 63x3x, Z of 0, so we're only doing one slice. This is channel 00. That's the Dappy one. This is the raw data from the Dappy one. Why don't we see anything? It's black. Okay, all you out there who play with Photoshop and other things, we took a 12-bit image. Windows Media Player doesn't know what to do with a 12-bit image. It cannot show you a 12-bit image. So therefore, we see black. If, however, I go to one of my snapshots, there's my snapshot. I do see that in full color because what's a snapshot? It's an 8-bit snapshot of the screen. So we can see that fine. So anyway, that just tells you what we're doing bit depth wise. And those are the pictures that we saved. All right, let me get back to the software. Okay, so for the moment then, that is how we get a decent two-dimensional single slice image using all of our spectral capability, well, most of our spectral capabilities on the microscope. There is a lot more nuance to this. This is the basics, but it gets us going, it gets us started. There's a lot more detail I'd like to talk to you about. We are out of time, however. I see in the chat that there are some questions, so I'm going to take those first, and then we'll go live with other questions. What I'm going to do with these pictures that I just took is we are going to post this recording on the BCA website. So if you want to rewatch any of this or watch it before next month's as a refresher, you can do that. We'll also give you a link to some of these, or we'll put these pictures up as well so you can see what we did. All right, so with that having been done, I am going to stop sharing my screen. And I am going to come down and take a look at the chat. Okay, we can make this as an exact link. Okay, so Catherine Hanlon, oh, hey, how are you? Asked, what is the exact microscope model we are looking at? Again, this is, yeah, you got it, Leica SP5. So again, this is the Leica Scanning Laser Confocal Microscope, SP5 2, actually. We got this system in 2012. The SP8 model came out just after that. Our second confocal that we're going to be working with next month is going to be the SP8. And just last year, Leica came out with the Stellaris series, which takes everything that I'm telling you right now and throws it out the window. So maybe in a few years, we'll have one of those. I have no idea. Michael's saying, okay, why mix? So Howard is asking why we mix detectors. So again, what we're doing is we are picking the detectors that are best for the sample that we're looking at. That's a simple way of looking at it. There's several ways to answer this question. The first way actually has more to do with money. I have 5 detectors on here. So theoretically, I could do 5 simultaneous channels at once. But as you saw, it's difficult to do a lot of simultaneous channels without getting some overlay. So we're probably not going to use all 5 at the same time. So I can kind of mix and match which detectors I'm going to use. Now, why PMTs? Well, sometimes the PMTs, like I said, are more useful, as in the case of the Dappy, because it's a lower contrast detector, and it gives me a better tonal range. The high D detectors are better for their sensitivity, but they're higher contrast. The PMTs, the standard detectors, are $8,000. The high D detectors are $45,000. Okay, there you go. There's a good reason. So I have 2 detectors that are high D detectors because that's $90,000 right there. And to add a third one was going to be a little pricey. So we go with 3 PMTs. They have to be placed on the detector in a particular order, so they alternate them. PMT1 is a PMT. The second position is a high D. The third is a PMT. The fourth is a high D, etc. Without getting into the mechanics, my service guy could explain it better than I can, but that is how they're laid out. We use them in ascending order as far as the wavelengths go. So I hope that answered that question. Jim is asking, when viewing fixed versus live cell lines, is there a preferred method of fixation? Jim, I'm going to have to get back to you on the specifics of why, because otherwise I'm going to sound like an idiot right now. But 4% freshly prepared PFA, because if you use a regular fluorescence or regular formaldehyde, you wind up with a lot of autofluorescence. Autofluorescence happens because of the of that oxidation that I was talking about in the traditional formaldehyde constructs. So freshly prepared only. Fix your stuff and throw out the expert. Don't save it.
Great, thank you.
Okay. Before Jim, I'll ask you again. Or just
Jim has another question. Yeah.
Jim does. Just 1 second. I do. If anybody wants to leave now, you're more than welcome. Thank you for attending. As I said, if you want to tune in next month, we're going to take everything that we did today. We're going to start with this, and we're going to build on it into 3D and hopefully a little bit of live work as well. We're going to try to do live 3D of mitochondria next month. Anyway, that's what we're after. Thank you for attending. Howard, you asked, is the mitochondrial stain, in the mitochondrial stain view, the nucleus seems bright. Is this bleed through? Yes, a little bit. Well, no, actually, I take it back. It is not. It was before we separated, but remember when we went to the sequence, the Dappy is not being excited at all. So when we're in that final image, when we were looking at that final image, we were seeing the mitochondria being excited with MitoTracker, from the MitoTracker red with the 561 laser, and the 488 laser was also on, but the Dappy laser was not. And so the 488, my lowest laser, cannot excite Dappy. It's not going to. So anything that you were seeing in the background may have been a little bit of overlap with the 488 signal, but not with the Dappy. However, there is that thing that I was talking about before about autofluorescence. And these particular slides are now about 6 years old. And so I'm getting a bleed through type effect because of just the age of the slides and the dye sort of blending out a little bit. So we're getting autofluorescence, and you could kind of call it bleed through from that. Okay, so hope that answered that question. Anybody else have a question you'd like to ask?
I know this probably varies with the number of slices, Jamie, but that last image was so cool. Like, what's the data acquisition time to get all those different slices, interface them into a 3D image?
Well, everybody, I hope you listen to that question, because that is what we're going to answer next month, and it's going to take an hour and a half.
Okay.
So there's no quick answer to what you're talking about. The challenge, as I said, this entire process is a balancing act. Everything that we've been doing today had more to do with quality and making sure that we're capturing the data, and very little to do with speed. But once you start doing stacks and you're focusing through many, many layers, you can have 100 slices in a stack. And if every slice takes a minute to scan, 100 minutes to take the image, that's not practical. So speed becomes a major, major issue when you're doing 3D stuff. And so we're going to talk about all that and how we work with that next month. So yeah, so that's a tricky wicket. And again, that's why no two sessions are going to be the same. And when you're doing with live cells or you're dealing with fixed cells, they're not going to be the same either, because there is something else called phototoxicity. So I got dead cells here. They're fixed, they're stained, they're not going anywhere. I might have a problem with bleeding, or excuse me, with fading and quenching, but those cells are already dead. But if I'm looking at a live cell, think about this for just a second. All right, many of you work in labs or in a hospital setting at least, and understand that there are hoods that the researchers use, right? And underneath the hood, there's a light that they flip on at the end when they're done to kill everything in that hood. And what is that light? Well, that light is a UV light, isn't it? So they turn on the UV light to kill everything. All right, so I'm using a UV dye here, Dappy, right? Now, I'm not exactly using 365, but doesn't it make sense that if I use a low wavelength that's down around the ultraviolet level, those cells are not going to be terribly happy being hit by that? And so you can take that and extrapolate it out. The bottom line is that the intensity and the wavelength of the light are going to have a negative, very negative impact on the health of the cells. And you have to take that into account when you are doing the imaging as well. So sometimes you can't get everything. You know, you can't get the quality, the speed, the live, the whole bit, you know, the stats to do that. Okay, so let's see. Randy, you are asking, do you collect data? I think there's several things, so I'll just do the first one first. Do you collect data for researchers if they request it? Yes, we do. So in our core facility, we basically do it several ways. We can either teach you to use any of the equipment here, or if you want to drop it off, we have services. So a core facility is essentially a small business inside of a larger one. The Institute itself has its overall budget, and we have ours. So there is chargebacks, and I think you find that pretty much in almost all core facilities anymore. When I first started doing this, you didn't, but I think it's pretty much that way everywhere. So in the core facility, there's a chargeback, and our chargebacks are based on instrument time and assistance time. So in the case of training on the confocal, for instance, you're paying for the confocal microscope and you're paying for my time to train you. But once we're done with the training and you're using it by yourself, you're only paying for the instrument time. Now, if you want to give me some stuff to image for you, or you want me in the room with you when you're doing your work to help you with it, sure, we can do that too. But again, you're going to be paying the full boat for both of those. We do have different rates depending on where you're from. So there is a standard rate, which is the academic rate. So we have people that come here from University of Pennsylvania, and they'll use it and work with us, and that's sort of the standard rate. Then there's a discounted rate for Wistar, because we're subsidized, obviously. So Wistar rates are lower. And then we also have a higher rate for commercial. So we do have some commercial users that will come in and they get charged an awful lot more. but we can do it with them. So, and again, they can drop it off if they want as well. So we have all three. And so that probably answers your next question. Do you charge for the training when you provide it? So that answered that. Do you train every student or postdoc who would like to use the microscope? We train anybody who gets sent down. So if a PI comes in and they have a student or a postdoc, or even they want to get trained, sure. The billing goes to a specific grant. So whoever comes has to know what grants they're allowed to charge to. And we use a system called iLab. I don't know if you've ever heard of that, but it's an online calendaring and billing software. So when they sign up for time for the scope in iLab, they have to choose a grant number. And it's hooked into our accounting offices here. So if you as an individual go in under your credentials, it will only give you the grant numbers that you have available to you. And if there's no grant numbers there, you can't sign up. So if there's a grant number, then you can choose. I'm not allowed to choose it. You have to choose it. But it really depends on the PI. Some PIs don't want everybody in the lab trained. Other PIs do. In the old days meaning 2 years ago, pre-COVID, I would train people together. And so sometimes it actually worked well to train 2 people at a time. So if I had 2 people from one lab, they both be in here, the training would take a little bit longer. So yeah, I wind up charging them a little bit longer for the training. But they could kind of bounce off of each other. One forgot, the other remembered, and vice versa. And it kind of created an effective, one person out of it that way. We're not back to full COVID, pre-COVID situation yet. We actually will be on July 1st. So on July 1st, we may go back to that again. But I'll tell you this, what I've learned in the last year is this method of training has worked really, really well. I didn't have the videos before. I had to create the videos specifically for this. And that takes up a good chunk of the time that we used to spend live in person. So now they get it from the videos instead. And then, so they have that as a reference, and it takes us less time. When we go live, I'm training from my office, right? So I'm sitting in my office and they share the screen, just like I was sharing the screen with you before. But when we're one-on-one, we're using Microsoft Teams, and I can have a mouse and they can have a mouse, and we can share the screen. I can point to things, I can click on things. And so we run through the entire process. Now, the advantage to this is they're doing hands-on from day one, right? So remember the way I explained I did it before, day one, they never touched the thing. But under the new way, they're doing it from the beginning. They're touching everything. I'm in my office. And then on day 3, they're continuing to do it that way. But now it's only day 2 now. So it now takes only 2 days instead of 3. They get more reference, so it costs less, saves them time. They have more reference from the videos, and we record the sessions. And I give them the link to the session so they can also re-watch the sessions for anything that they might have forgotten. And I've had people just 2 weeks ago doing that. So all in all, this method of training has actually worked out very, very well. So I'm going to continue to do this, but I think that we'll probably allow 2 people in here again once July 1st hits and we go back to open mask and everything. Yeah. All right, let's see if I answered that question in your last question. When you train new users, do you have them provide their samples and help them set up the acquisitions optimally? That is part of the last day, yes. So usually what we do is, remember, training is training, right? The idea is to get you to learn how to use this thing. And so on the last day, which used to be 3 and now is 2, we usually go over the 3D aspect, the stats. Even if that's not something that they're always doing, it's something that they might be doing later. So we're gonna cover that. And so when we're done with doing the 3D stuff, then we go to their stuff. And yes, everyone is different. They're gonna come in with a dish or a slide, and we're gonna put it on and immediately realize the problems. And we're going to work out the solutions together to make sure that we get everything optimized. It's very important when you do that that actually it's best as a PI or as a researcher to come and talk to us first, because let's do a simple thing. One of the things that happened years ago, before I had the SP5, I had the SP2, we didn't have a laser on that microscope for Dappy, right? The 405 laser was an add-on that wasn't purchased, and the scope was purchased before I came here. So I never had a Dappy option. So people would want to get trained. They'd come down, they'd have a slide with Dappy and 2 other colors and say, let's get going. And I said, this is great, but we can't see the Dappy. And they'd say, why not? Well, we don't have the laser. Well, I didn't know that. Well, you should have talked to me first, right? And then you would have known that. And there are other options. So there's things like at the time, ToPro 3 is a far red option. So we have, which also labels DNA. So there's always other options. A lot of times people will come down with a dish that doesn't fit right. Or they'll have a dish that is not a cover slip bottom dish and want to do high magnification, high resolution stuff. And with a regular plastic dish, you can't do that. You have to have a glass bottom dish. So they might have done all of their work in the wrong dishes. So it's always important as a researcher to talk with the people that are handling the instrument first to make sure you're not doing something that could easily be avoided. Make sure that the experiment makes sense, that you have everything, that you're approaching it the correct way. And then also it's very helpful for us to know what you want to get out of it. Are you trying to do quantitation? Are you only trying to take a nice picture for a cover? What is your purpose here? It used to be that I would tell people, well, depending on your purpose, you either do 8 bits or 12 bits. I don't say that anymore. I tell them just do 12 bits, period. Because you can always resample down to 8 bits, but you can never upsample to 12. You'll never get the data out. So, so there's things like that. I could go on and on. There's lots of examples like that. But yes, so like I said, the last part of the training always covers what they're talking about. All right.
Great job, Jamie. Yeah. In the interest of time, if anybody has any more questions for Jamie, you can email him. Jamie, why don't you put your email in the chat, bca.org. That's it.
So this is my BCA website as VP, so you can get to that anytime.
Terrific.